All right, now in the first hour, let's summarize what we have learned. We have learned that we do sequence alignment to find the biologically most possible way that these two homolog sequences are related to each other. Find the matching pairs, find the pairs that are inserted, deleted in the biologically most possible way. Now, and I have shown you that this is used, I have talked about wow, in different places that this may be used to infer function, to find conserved regions, functionally important parts, to assemble sequenced individual sequenced DNA against the reference genome, and or even for novel sequence assembly. Okay, in novel assembly, again, we may need to use alignment of sequences because the only way to build a jigsaw puzzle without a reference picture is you have to figure out a way how these two jigsaw pieces fit each other, right? I mean, if I give you 10,000 jigsaw pieces and I want you to solve this jigsaw puzzle without looking at a reference picture, you will try to just find some similarities between two in terms of color, in terms of connecting regions. You have to figure out a way to connect two jigsaw pieces together. And in novel sequence assembly, these short sequences that are sequenced by these machines, actually they contain overlaps. Without these overlaps, we will not be able to we can just a random permutation of these sequences truncated one after another will be just an assembly. Any of them will be a correct assembly as far as we are concerned, because when there is no overlap. These overlaps are found with the help of, again, alignments and the graphs that again mentioned in the first hour, the De Bruijn graphs, are actually they find these overlaps between sequences, the overlap between the suffix of a sequence and the prefix of a sequence. Another sequence can be used to connect them together with an edge, with an arc, a directed edge, which we will help these graphs, and finding a nice long path on this graph will indicate probably a solved jigsaw puzzle. All right, so this is sequence alignment is used in a lot of places. Now, I already mentioned about the motivation, so I'm going to skip these parts quickly because I mentioned that DNA sequences are related, and via evolution they're related to each other. And after alignment, we can find important regions that are conserved. We can use alignment to determine function, to uncover different evolutionary forces. So it's used for many, many different things. So this is the motivation of why we use sequence alignment. And it's the procedure of comparing two. When it's called pairwise alignment, it means we're aligning two sequences, pairwise sequence alignment. And in multiple sequence alignment, which we are going to see later, and since the algorithm that we see this week, the dynamic programming, does not scale well to the number of sequences, because although for pairwise sequence alignment, we are going to describe an efficient algorithm with polynomial time complexity. When the number of sequences gets larger, the algorithm will be computationally exponential in the number of sequences we align. Okay, so that's why it's not going to be feasible to multiply align multiple sequences together. And it's basically mapping, providing a one-to-one mapping between these two characters. And we are actually observing certain constraints while we are doing this one-to-one mapping. For example, when I align a tryptophan here and to another tryptophan here, these W's, okay, they are aligned with each other, they are matched. I cannot match an amino acid here to another amino acid here. So these matchings cannot cross each other. When I do a match, I should just, I can match, after this first part is matched, for example, the remaining part here, the remaining amino acids that are on this right hand side, they can be matched only to amino acids that are on the right hand side if these two are matched. So this restriction actually helps us a lot in defining our dynamic programming solution, because this, for example, if A and A is aligned, the next step is one of the three things. Either H and D is aligned, either there's a gap. By the way, these insertions, deletions are called also, we call them gaps in the alignment. For example, if there's a dash, that dash is called a gap. If there's a gap against a dash, it means that H is inserted here or H is deleted here in the second sequence. So we can have a gap against the dash, against H here. We can have a gap against the D here, or H and D can be matched, okay? If we just consider, if these first two parts, let me just use the marker. Okay, now consider this part and this part. If these two parts are already aligned with each other, this is like starting an alignment. If somebody gives you that this first part is aligned, the way the second part is going to start is only one of these three things. It will either start as H against the D, or H against the dash here like this, or D against the dash. Okay, so one of these three things will be the first column of that second alignment part, right? I mean, can we have, for example, two dashes against each other after AA? What does this mean? A gap, another gap. So this doesn't correspond to any biological event when we have two sequences. This something is deleted in both of these sequences, but with only the information we have is about these sequences, it's impossible to predict they are both of the same amino acid is deleted in both of them. There is no evidence of such a thing in these sequences. So such an alignment column is nonsense. We cannot have something like this. All right. There, for example, they have both isoleucine deleted in this position. How do you know that? There is no evidence about that here. So since we are trying to do something biologically most plausible, having two deletions in both sequences without evidence of something inserted there in some other sequence, the existence of that in some other sequence would not make any sense. So these are the only three possible things we would have here on the right hand side, since we have this H cannot be aligned against something W or on the left hand side. Okay, so if this is given, therefore it will start with like this. Okay, if you say, okay, I can maybe align H to this G here. Okay, if you align this H with this G, it's doable. What does it mean? It corresponds that you should start your alignment with this D gap. If you want to align this H with this G, it means that in the first sequence right here, you have a lot of gaps, all right? So, or not maybe a lot of gaps, but this D has to be aligned against the dash, so this H moves to the right, okay? Also, actually, there will be a lot of gaps. There will be exactly 3 dashes. For you to be able to align H with a G, you should have this, dash, 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 and D, V, A underneath. Only then you could align that H with this G, right? And this is one of the possibilities we already consider here. So we consider all the possibilities. So this restriction, this constraint, will allow us to formulate this dynamic programming solution easily, because this makes the alignment process additive. In other words, it's like we can define a recursive solution. If somebody gives you a partial solution, you'll be able to add up onto that partial solution by build upon that partial solution by considering the next possible thing that can come after that. Just like we did here for finding the number of possible alignments, right? When we were finding the possible number of alignments, somebody gave us what pi minus 1 pi, these 3 cells. If you know these 3 cells, if these are given to you, it's really easy to compute this entity. Yeah, so here the formal definition of sequence alignment is that given two strings, pairwise sequence alignment, and this is, as I said, global pairwise sequence alignment. In global pairwise sequence alignment, we map all of the characters in either of these sequences have to be matched against something. We cannot just say, I will do the alignment only for these four in the middle. I want to just align these four and ignore the remainder of the sequences. That's called local sequence alignment, and we're also going to see that. And it's like finding the best local sequence alignment is. It means that you're free to ignore the beginning of either sequence, the ending of either sequence. You're free to ignore those and find something that are in the middle of both sequences like this. For example, you may align this part with against this part. They don't have to be underneath each other. This part may be aligned to this part here, and that will be a local sequence alignment. And given these two strings of length m and n, an alignment is an assignment of gaps to positions in one string and the positions in another string so as to line up each letter in one sequence with either a letter. So everything has to be mapped against something, either a letter or a gap in the other sequence. So that's what a alignment is. This is a definition. And when we talk about the best alignment, we need to talk about scoring. Okay, now let's talk about scoring matrices. And I talked about homology. So yeah, so the term that I forgot the name in the first hour for the hemoglobin, I just stopped in the mid-sentence if you had noticed that. They're called, and I just froze like that. They're called paralogs. I forgot this term. The hemoglobin, different versions of hemoglobin alpha 1, hemoglobin alpha 2. They are, if they are within the same organism, they are like the same gene duplicated within the genome. If this divergence of at certain point in evolution, that part of the chromosome is duplicated and started appearing in two places in the coming generations. When we have these two places, those two genes, they also undergo different changes. They start to diverge. They start to differ from each other from generation to generation. So this is what divergence means. There are insertions and deletions or mutations specific to each copy, because these changes are like, they may sometimes be local. It means that if one copy is updated, it may not be affecting the other copy. So they diverge from each other as time goes on, as the evolutionary forces take action. So duplication means if divergence follows the duplication within the same organism, it's called a paralog. And if this divergence happens after the speciation events, if these two homologs are in two different species, like mouse hemoglobin and human hemoglobin, are called orthologs. So this is like the more general term, the term at the end. All of these are homologs. Okay, these are more specific terms that describe homology. Or when you talk about these are ortholog, oh, you understand that, oh, they are homologs that appear in two different species. Or when somebody says they are paralog, they're homologs. They're on the same organism, same genome. And there's also xenolog. I forgot what it means actually. So it's probably not so important for this course, introductory course. But if you take, for example, an evolutionary biology course or phylogenetics course, you will have to go into more details and understand all these terms in much more detailed manner. But for us, they're just sequences. They are related with each other. Okay, like these. There's a speciation with that, the globin family. There's in certain organisms there is only one globin. All right, but after this there's some gene duplication that happened in evolution, and all the organisms have alpha and beta globin. On this side these are the alpha globins. So let me start annotating again. Alpha globins and beta globins here. Okay, so a frog alpha globin is a paralog of frog beta globin. So again, I'm not going to go into details of these. You just assume that they're functionally related. Now, so the evolutionary relationships and understanding these evolutionary events is our goal when we do sequence alignment. And there are different reasons of variation between different organisms' sequences. There may be substitutions. Nucleotide substitution may be due to some replication error or some chemical reaction. Certain C's may be changed to G's, A. So we may have these polymorphism. We have that a lot actually. Humans have these single nucleotide polymorphisms, changes in certain parts of the genes is in single nucleotides that appear. I think the threshold was like at least in the 30% of the population. So there is this single nucleotide variation when it's called a polymorphism. It means that it's actually observed in the population a lot. It's not specific. That mutation is not just specific to a single individual. We see that in a lot of people. In at least 3% of the population have the same variation in that single spot. So what does it mean? It may be correlated with having blue eyes. It may be correlated with having diabetes type 2. So that's another thing. These variations, how they're associated with these diseases, or the area that studies these variations and their relationship to diseases is called genome-wide association studies, or GWAS. Okay, I'm not going to go into GWAS studies in this introductory course, but there are courses in the Health Informatics Institute, Bioinformatics department that details all these things. So insertions, duplication, and they have different rates of DNA evolution in different parts. Certain parts have a higher rate of observing these changes, different parts less. So it's not uniform, but all these knowledge that I give here, different rates of evolution, different parts changing differently, when we make these assumptions in our sequence alignment, we will make these incorrect biological assumption to have a mathematical model. We cannot model the entire nature in 100% accuracy when we're trying to answer these questions. Therefore, to have some viable practical solution, we make assumptions, obstructions, certain things that may be wrong, like all nucleotide sites. A mutation somewhere does not affect the mutation in another place. We make this assumption in our scoring function, which may be not true biologically. The substitution rate is constant over time. The base consumptions that equal all these biological statements that may be not true. We will make these assumptions in our sequence alignment. Okay, which will lead us to things like the cost of a mismatch. If you mismatch a nucleotide with a different type of nucleotide, the cost of a mismatch is -3. And we will take this as -3 throughout the entire alignment, throughout the entire sequence. Or with being amino acid, we are going to assume that matching this amino acid to this one has a positive score of 6. And we will take it as positive score of 6 throughout the entire alignment. Again, which may not be entirely true, but these are what we need to do, as I said, to have a practical working algorithm. It's impossible. Sometimes simple things may lead to still promising results. And this is what we do here. If you complicate the thing, first of all, we may not have the knowledge to actually use what the actual values are. For example, you're saying that the match here is 6. You may say, okay, the other part, you should not use the same match score as 6 in the other part, but what am I going to use? How do you know whether 5 is better than 6 in this other part, or 7 is better than 6? So you need to have some additional information to actually promise something better. And most of the time, we don't have that knowledge. That's why we make these simple assumptions. So I told you before that if we are looking at different alignments, in order to figure out which one is a better alignment, we need a scoring mechanism. One way to score these things in the nucleotides, actually, nucleotide DNA sequence alignment, we usually have fixed match, mismatch, and gap match score, mismatch penalty, and gap penalties. For example, we can just say this alignment has 6 matches, 3 mismatches. We just count them separately. Okay, so each column contributes to the score independently of the other columns, and we can just finding the score is just a simple means of counting the number of matches. This is a match, GG match, GC a mismatch, CG another mismatch, TA another mismatch. So I have 3 mismatches here, and the rest is 1, 2, 3, 4 match. So I have 6 matches, 3 mismatches, and there is 1 gap. So this other alignment has 7 matches, 1 mismatch, and 3 gaps. So it has more gaps than the other one, but now it has less mismatches. It's really, which one is a better alignment? Is it the one with the highest number of matches? Maybe. But what about, if we penalize mismatches differently than gaps? So why do you penalize mismatches more than a gap? Only if biology requires us to do so, which means that if in nature we observe insertions and deletions more compared to mismatches between homolog sequences, it means that having an insertion and gap is much easier in nature than having a mismatch at that position. So if that's what we have, this is what we hope observe among homolog sequences in nature, then we should probably give mismatch penalties a larger penalty and gaps a smaller penalty. All right, so this is how we are going to score these things, and this is how in 1970s and 80s the researchers that tried to define these scoring matrices did. They looked at families of proteins that are known to be related. They looked at their sequences. They looked at whether which amino acids are changed to which other amino acids more commonly, and figured out those match mismatch scores. Usually in DNA sequence alignment, we have a single match mismatch penalty. And another thing here is that the gap penalties. What we need to really differentiate gaps that are like this. Let me get a new. Oh, actually, maybe I can use my notepad here, but I can even increase the size here. So look at these two alternatives. Okay, and I may have another thing that looks like this. Maybe the same number of gaps. Okay, there are 5 gaps here. Let me also have 5 gaps here. So in terms of penalties, okay, they have the same number of mismatches. Same, maybe, I don't know whether I have 5 of them here. 1, 2, 3, 4, 5. Yeah, both of them have 5 gaps. Okay, same number of mismatches. Which one looks biologically more plausible is what we should ask ourselves. And in nature, we see that gaps, insertions, or deletions that span a certain number of nucleotides is more common than these individual insertion or deletions. So how can we solve this? Can we say, for example, if I have a gap penalty of -1, having a gap costs you -1 units in your score function. In both of these cases, it's going to be a total of -5, right? There's no way to differentiate between these two cases. I mean, your algorithm will either output this one or this one randomly equally likely. How can you make your algorithm output this one, not the one at the top? A single gap penalty would not be sufficient, right? We need to differentiate things like the first time you introduce a gap and then extending a gap. For example, introducing a gap may be like -3, but extending a gap may be -1. Give less penalty to an already existing gap. And in that case, each of them are introduction of a new gap. It will be -3, -3, -3, -3, -3. It will be a total of -15 penalty for the gaps at this sequence. But in this one, it's going to be -3, then 4, 5, 6, 7. It will be minus 7. Minus 15 compared to minus 7 is much worse. It will obtain a much worse score. So the algorithm, we guide the algorithm to produce this one, which is biologically more plausible. But you see what we did here, by changing the scoring function, we guided the optimization algorithm, because the optimization algorithm doesn't know about biology at all. It just, whatever scoring parameters you give to your algorithm, it's going to output the one that gets the maximum score. But that maximum score, if you define your scoring function in a bad manner, that maximum score may not be a biologically good result. So that's the importance of biological these scoring functions. This is, I just wanted to go over why we need the scoring functions. So good alignments, we are going to have gaps between them. The gap penalty is one. So this scoring function, the match, mismatch, and gap penalty, if somebody gives these to us, hopefully in this course, I'm not going to talk too much, especially in this first weeks. We're going to, I'm not going to talk about much about how we obtain these scoring values. Usually these days, when biologists align their sequences, they usually, there are existing widely used scoring parameters that are used all around the world. So instead of trying to figure, this is really not a problem. Which scoring function I should use is really not the problem the biologists face these days, because they're usually the default scoring function that are used by the tools today. They are trusted. They produce nice results. Usually don't play with them too much, because finding the best scoring function for a certain type of sequence match is something that it's a different problem. I'm not going to talk about this. So we will just assume that in this first week's, the pairwise sequence alignment, we are going to assume that a scoring function is given to us as an input to the optimization problem. Somebody tells us that matches are scored plus M, mismatches are scored minus S, and gaps are scored minus D. And the entire score, since this is the alignment itself is additive, we are going to compute the entire score as a summation of these individual column scores. And I talked about this more complicated gap penalties in this example. So this example that I gave on Notepad, the way that differentiates the starting of a gap and extension of a gap differently, that is called an affine gap penalty model. In the linear gap penalty model, each of these gaps are penalized separately. There is a single gap penalty, which will produce minus 15. If it's, for example, minus 3 for all the gaps, it's going to be minus 15 in both cases. In linear gap penalty, each gap is penalized individually. We don't distinguish between whether it's an opening of a gap or extension of a gap. And for the dynamic programming algorithm that I will describe, we will use this linear gap penalty for model for simplicity. All the modern pairwise aligners or database searches, they actually use affine gap penalties, because biologically, what we have seen is insertion or deletion of a long chunk of sequences is more widely observed than individual insertions or deletions. But as I said in this introductory course, for simplicity, we are mostly going to focus on a linear gap model. Scoring matrix, as I said, is important. So here is an example scoring matrix for amino acids. So this scoring matrix that you see here, this is not an alignment, okay? Most of the time, the students just confuse this with the table that I showed here. Look at this table, the sequence that is on the top, the sequence on the left here. So this is sometimes called the partial scores table, or the dynamic programming table, or the partial alignment scores table. It's entirely different than this matrix. This matrix is our scoring input to our algorithm. This basically shows that the match score of aligning A with an A is 2. The score of aligning F with an F is 9. the mismatch penalty of aligning C with a D is -5. So that this is a table of these M and S values, match and mismatch values for every possible pairing of different types of amino acids. Okay, so if we had the same table for DNA sequences, you would have a 4x4 table, right? I mean, the scoring function, if you want to give different matches, mismatches to different types of nucleotides, you would have a 4x4 table showing what happens when you match A with an A with a C, A with a G, or A with a T, or vice versa, C with an A. So, and it's a symmetric matrix. Therefore, it's just the upper triangle is usually shown here. So this is, it's called the log. So this is a scoring matrix named as PAM. So this PAM matrix is one of the most widely used ones. It's PAM 250. There are different versions of it because these matrices are optimized for certain sequence divergence levels. If you have two sequences that are very distantly related in evolution, you should probably use a different scoring matrix that reflect that. And another scoring matrix which is used these days a lot as by default is this Blossom 62 matrix. For example, if you go to BLAST database search, this is the matrix that they use. Again, this is nothing but a 20x20 matrix. Regardless of your sequence's length, whatever length of sequences you are trying to align, your protein sequences, this is an input to the algorithm that shows what is the score of matching an I with an I. Isoleucine with an isoleucine, you get a positive score of 4. But some of those, a tryptophan with another tryptophan, the score of matching those two is 11. So even some matches are valued higher than the others, and these actually reflect how common these amino acids are in nature. This tryptophan is a relatively uncommon amino acid found in nature compared to, for example, one that gets a score of 4, which means that we want our algorithm to value if these two other proteins both contain tryptophan at some position. Aligning these two positions on top of each other should be valued more because that's really something you see rare in nature. The algorithm first prioritize matching those parts. That's why we give it a higher score. And some interesting thing that you see here, probably if you've noticed this, some mismatches are scored positively. Look at this. I and an L, a score of +2. An isoleucine and leucine. In terms of characters, if you just ignore the scoring matrices and have a program that just checks whether those two characters at that spot are same, it's a match. If it's different, it's a mismatch. If you just use something like that, it should be a mismatch, right? But isoleucine and leucine actually is considered as something positive, a match by nature, because we probably, when homolog sequences are aligned on top of each other, seeing a transition, it's seeing a change from a leucine at one position to an isoleucine in another position is observed more than randomly. So this is what nature prefers, potentially, possibly. So in a biological plausible alignment, having isoleucine matching with a leucine, we don't penalize it. It's okay to have that in a column. Okay, so this is our scoring matrix for proteins. We are going to talk more about these things when we come to talk about profiles of sequences. We'll see how they are actually computed as log odds matrices. Log odds basically means that it's like 2 times more likely an isoleucine leucine a match. 2 here is like 2 times more likely to see in that position than a random matchup. And they are built from some known homologies, and there are some differences between these two scoring matrices. Again, I'm going to come to those later. But a typical scoring matrix that is used today is to use a score of match 1 for DNA sequences. These are the scoring matrices that are used for protein sequences. Blossom 62 is used a lot. A gap opening penalty of -11. So initiating a gap is penalized a lot. But when you extend an already existing gap, you just add -1 to your penalties. Now, in the last 10 minutes, I'm going to now talk about the dynamic programming solution. I'm going to give the recurrence relation, the solution to this pairwise sequence alignment problem, and then how we can use this recurrence relation to fill in this table. This is the same matrix that I showed on GIMP, on my whiteboard. A path here corresponds to an alignment, and the number of all possible paths, which I already did actually, is exponential. So we cannot look at all possible paths. Now, what can we do? Here we are going to be using, if we are given two sequences, length m and n, if the first I characters in one sequence is aligned to the first j characters in the other sequence, okay, if this part of the alignment is already given, we are going to know that in the other sequence, we are going to start at I plus 1 and j plus 1 here. And whatever, if somebody gives you the aligned results of these two parts separately, the total alignment score that you compute is additive. This being additive is a little bit tricky, especially for affine gap penalties. For example, if this last position is a gap, this will not be entirely true. Okay, if the last position is a gap here, if it's followed by a gap here, the total number of penalties is not going to be like that. We need to combine them in a different manner than this one. But if assuming linear gap penalties, whatever the score you obtain for the first I and j characters, you can just add up the scores. The score is also additive. With that, we can actually extend this. We are ready to define a solution to our problem. Here, let me just, I will just use the board to show you the dynamic programming solution. So if somebody gives you the first I characters, so this is index I in one sequence. I don't know what this ends up with, but imagine the first I characters is aligned with the first j characters in the other sequence. Let's define the result of the optimum score, the best score that you can obtain to align this first I and j parts. Let's call it ij. Okay, Fij. I can write it like this. Imagine I have a function. So I define a function, the best way to align the first I characters to the first j characters of the second sequence. Now, what I can do is, if you want to add up, for example, if you want to grow it to one character more, okay, or if both sequences one character long, if you want to figure out what is the best way of, actually I should have started maybe I minus 1, j minus 1, but it doesn't matter. I mean, just the indices are just one more than the solution that is actually shown on the slides, but the idea is exactly the same. So imagine our goal is to figure out what is the best way to find out how the first I plus 1 characters and j plus 1 characters are aligned with each other. Okay. Now, if focusing on this last position, the first I plus 1 j plus 1 characters. So, for example, maybe the sequences may be like this. For example, A, C, G, and this last G may be. Let's assume this last G is the I plus first character, and here I have another sequence. Let's call it TACC, and this C is the j plus first character in the other sequence. Now consider and I'm going to align the entire first I plus one characters to the entire j plus one characters in the other sequence. Now this is what I'm trying to figure out. Now let's look at the last column of the alignment. Last column of the alignment that aligns the first I plus one and j plus one characters. How does the last column of the alignment look like? Is it like this? The first j plus one and I plus one and j plus one characters. So this is the last column of the alignment. Is it, should it be, I mean, is it for certain like this? No, right? It can be, the last column could be like this, or the last column could be like that. Again, if it's like this, the C has to be, the j plus first character C could appear before the last column. So if this is the last column, if we have a gap against this G here, this j plus first character C should be treated earlier to be able to have a gap here in the last column. Okay, so the last column is one of these three things. So what I mean is that the best way to align these three things, so it's, if I go over to the slides, so yeah, this one is showing actually here. So where do I have, yeah, the last position. So if you look at these things, so this is this actually starts talking about looking at the first position, but the idea is the same. So if these two, if you consider the sequences CA, CGA, and CGA, the first position can either be this one, a gap against the second, or a gap against the first. So these are the three possible cases, and each of these three possible cases comes with some score of penalty if we make that choice. But the remaining sequences become like this, and so this actually kind of an analysis makes it difficult to compute this one recursively like this. But what I'm going to say here is that it's going to be either, if I want to figure out this one, this option, what does this option mean? This option means that if you, these are the three possible cases the last step of the alignment look like. So what does this option mean? It means that a mismatch penalty of GNC, right? If you take this option, you have to pay the mismatch penalty. In other words, for this option, I'm going to take a look at this. A mismatch plus what? What is the score of the best way to align the first ij characters of the sequences, right? I mean, if G and C is matched in this last column, you have to know the best way to align the first I and j characters in the other sequences. So if somebody gives this to you already, and if you choose to use this as the last column of your alignment, your total cost of this option will be this, right? A mismatch of G and C plus the cost of whatever Fij is. Okay, now this is one option. Is this the best option? I don't know. And I don't need to know actually. All I need to do is I will just compute what happens if I choose this option. Just can you tell me what will be if I choose this option, the second option? So let's label them 1, 2, 3. If I choose the second option, what is my total cost of the alignment? What is this? And assume linear gap penalty. If I have a linear gap penalty, this last column will come with a gap penalty on its own, right? And what should I have on the left-hand side? If this is the last column here, this is the last column of the alignment, what does the first part look like? How many? It's going to be Fi minus 1. Wow, we have a thunder strike here. So it's going to be Fi minus 1 j, right? So you probably have seen the flash of the lightning as well. So it was really loud, but it was still at least 1 or 2 kilometers away. So this is going to be, since G is the, so not I minus 1. I actually did something wrong here. Can you correct me? It's going to be I, and this is going to be J plus 1. Okay, I forgot that I'm using this I and J, this G and C are the I plus first and G plus first characters. Now, if I have the second option, this is for the second option, this was for the first option. For the second option, if I choose this option, it means that the J plus first characters are already on the second line. All right, so somebody should give me the best way to align the first I characters of the first sequence against the first J plus 1 characters of the second sequence. And the third option is gap plus F I plus 1 J. Okay, so this is the third option. If you use the third option as your last column of your alignment, you again need to pay a gap penalty, and you need to know the best possible way to align the first I plus 1 characters, because you want to have G appear on the left-hand side. So that's the I plus first character. So what is the best way to align the first I plus first characters against the first J characters? Because to the left of the C, I have only J number of characters. So these are my three options. Which one of these three options is the best one? I don't know. So how are you going to select the best of these three options? We have a nice mathematical function for that, right? Max. Look at all three options and choose the maximum of these three ones as your optimum way to align these three sequences. So what I have written here, if I plus 1j, I plus 1j plus 1 is equal to the maximum of some match or mismatch penalty. This may sometimes be the match score if the two characters are same. Okay, it will either be a match score or a mismatch penalty, and whatever is the best way to align the first ij characters. And this will always be a gap penalty if you choose these two options, and these are the best ways to align. So this is what I had written on this whiteboard, is called a recurrence relation, and it's actually the solution to the pairwise sequence alignment algorithm. If you solve this for f and m, okay, if you have a sequence of length n and a sequence of length m, if you ask for this, it will give you, by writing these equations, f and m is going to be either, so what is f and m? It's going to be the maximum of these three things. Maximum of match or mismatch, depending on whether the last characters are same, match or mismatch plus fn minus 1, m minus 1, or gap plus f and m minus 1, and or gap plus f n minus 1 m. Okay, I will look at these three choices. So how do you figure out what f and m minus 1 is? Recursively solve for this until you get to f00 or some other base condition. You can solve this recursively. Again, if you implement this as a recursive function, it will take forever to run. That's why we use something similar to this one. We are going to, in order to compute this f and m, we are going to start building our table starting with f00. What is the best way of aligning 0 characters from the first sequence against the 0 characters from other sequence? So the score is really 0. Okay. Now, let me ask you, before I stop today, let me ask you this. For example, what is something like this? What is f0j? Which means that it's a cell that is here. We filled in with all ones here. It was all ones here. Now, assuming a linear gap penalty, and the gap penalty is like minus g. If the gap penalty is minus g, and this is the jth column here, what am I going to write here? Something below 0, exactly. But if I give you these numbers, for example, the jth character, and a single gap cost me minus g penalty. How many gaps do you think you have in order to cut? Exactly, it's not 1 minus, actually exactly 0 minus j times g, or if you end up here, it means that you have j gaps. This one is also aligned against the gap. So the actual correct solution is going to be minus j times g. But you have found solutions that are very close to that. Since we start at 0, this one will be minus g, minus 2g, minus 3g. Oh, actually, you're right. You may say j minus 1 if j starts at, this is column number 1. If you start your column numbering at 1 here, what you wrote on the list perfectly correct. It's going to be j minus 1 first column here, but we usually label this first column and first row as 0. So this, we start at numbering from 1 to 3 here. So this will be like, for example, if this is a 5 character in the sequence, there will be exactly 5 gaps introduced here. We come to the 5th sequence. Therefore, it will be exactly 5 times minus g. All right, so we are going to fill in this first column and first row here. And how are we going to fill the remainder of these cells? By using this equation that I wrote here. You just take a look at, in order to fill in a certain cell, what should I need to know? This one, and this one. And if you look at my table here, or any table, for example, if you look at the table here, in order to fill in this cell value, all I need to do is look at this value, and this value. Am I going to this time again add these up and right here? No, I'm going to add a gap penalty to this one, and take a look at it. I'm going to add a gap penalty to this option. So this is my gap penalty options, and this is the option which is either a match or a mismatch. And I'm going to compare these three. I'm not adding them up or doing anything. My mathematical function that I use to compute the cell value here is that max function. I'm going to look at, am I choosing the maximum of these directly cell values? No. This is also really important. I'm adding a match or mismatch penalty to the cell value here, then compare it against a gap penalty added values of these cells. Okay, whichever is maximum, I write it in this cell. So filling in each cell takes 1 time, which means that in constant time, I can fill in an entire cell. So it's just similar to this one. In O&M time, I will be able to figure out the best way, the optimum way of scoring these two sequences, aligning these two sequences, and I will know the score. But we just don't want to find the score, right? We also find what the alignment is. And we are going to come to that later. Sorry, I took 9 minutes of your additional time. So I'm going to stop recording here. I'm going to continue on